me try. Good. It's my pleasure to welcome us once again, and I am my humble self, Dr. Ashraf Akintola. So today we'll be going through the how to conduct genomic analysis, and I'm going to take us an important tool that is used in that regard. So the tool that we'll be talking about is the Galaxy. So Galaxy is a tool that is a very, very user friendly and can be used for a myriad of all analysis. And in that case, I'm going to be sharing with us my screen so that we all can see how it's been done or what it's being used for. For that, I'll be sharing my entire screen, and I would love that we also take part as I take us one after the other. Forgive if I'm jumping into the process right away because I don't want to waste any of our time any longer. So the tool we'll be using is called Galaxy, and for those who are familiar with Galaxy, they would know that this is a tool that is very readily available and it is free to use across all the anywhere you have, you can use Galaxy, and you can also use it to perform different analysis. But for the sake of this class, we'll be using it to perform the analysis of genomics. So to start with, Galaxy is assessed either through the EU standard or the EU platform or through the African Galaxy platform. And for that purpose, I'm going to be focusing on the Africa Galaxy platform, which is the window that is responsible or that is being used by African scientists or dedicated for African scientists use. So the website is africa.usegalaxy.eu. Africa.usegalaxy.eu. If you are with me, are you with me? So please let me know if you are with me. Are we together?
Yes, sir.
Yes, sir.
Thank you so much. So if you are with your laptop here, I want you to go to africa.usegalaxy.eu. And if you are not with your laptop, I want you to type into your phone. You can minimize the screen for the Google Meet. So just go to africa.usegalaxy.eu. Now, once you go there, it will take you to the platform whereby you would have to start your journey in bioinformatics. Now, the reason why I'm doing this is because this class is supposed to be an hands-on, and for the purpose, I want everyone to be able to get one or two things out of this sitting, and you should be able to perform a genomic analysis irrespective of your level of expertise. And I'm going to show you how to do that within few minutes. I've wasted enough of your time already, and I'm going to make you jump into that process as soon as we have it. Now, I've already registered my own account on Africa Galaxy and .eu use galaxy africa.usegalaxy.eu. I've already had my account registered, but if you are just starting out, you can see that here uh, you can if I sign out, I'll have to sign in again and do a lot of another new process. But for the purpose of this class, I wouldn't be showing us the process. So if you are just starting out, you have to create an account. And once you create an account, use an email that you have access to and register, and then a confirmation will be sent to your email, and then you would have access to this platform to be able to do a lot of analysis that you want to do. And in that case, after you have gotten the analysis, your account opened, so use African Galaxy. Galaxy will give you about uh, 270 gig, about 270 gig of data that you can use for your analysis. And this is one of the peculiarities of you having being a bioinformatician. Bioinformatician, by definition, is using computer software to analyze biological data. So you are converting biological data to a language that computer will understand, and then which will now be converted to an analysis that will give us an information that will be useful to whatever it is that we want to use it for, either for plants, for animal, for humans, for bacteria, for fungi or virus. So those are the things that you can do as a bioinformatician. But this class is dedicated for genomics, and genomics is understanding the language of your DNA, your gene. You are understanding the language of your gene. You want to decipher the information that is inside your gene using using computer languages, computer programs to decipher the information in the gene, either in humans, in plants, in bacteria, or any other thing that is having a gene. So that is what genomics is all about. Now, there are different places or key factors you need to understand about Galaxy. Galaxy has different interfaces. So one is this Galaxy home. You have upload, you have tools, you have workflows, you have workflow invocations, you have interactive tools and histories and the likes. You have different interface to interact when you are dealing with Galaxy. Now, what we want to be, I want to show us, or I want to give us an insight into how to perform genomics analysis. So we are going to go to this particular place whereby you have this tassel on your graduation cap, which shows something relating to university education. So you can click on it to see the training manual. Galaxy has a wide array of different trainings for different purposes and for different uses. So it depends on what you want to use or what you want to focus on that will determine what you're going to do with Galaxy. And there are different data that you can also play around with that would make you become expertise and be able to use that expertise on your own data. I mentioned earlier on the importance of storage. Storage is very, very important because one of the things that you'll be doing in bioinformatics is that you'll be accumulating data, storing the data, and then interpreting the data. One of the challenges that we have as African scientists, however, is that we do not have the appropriate resources to have abundant databases to store all this data that we are generating. Nowadays, we have a lot of data being generated, but where are we storing the data? Where are we keeping this data? So, and that is why it is important for us to have enough data. And that is one of the things that Galaxy offers you free of charge. You are not paying for it. It's as having a dedicated laptop with this particular data volume that can be useful for your work. So now we've clicked this particular icon, and then you have different things that you can work with. So one is introduction to Galaxy analysis. Second, using Galaxy and managing your data. Now you have different scientific fields. You have climate, you have computational chemistry, you have SARS-CoV, you have foundation of data science, ecology, data science, genome annotation, and the likes. Now, as it is said that genomics, I'm going to take you through a step-by-step process on, I will assume that, yes, we are complete beginner. Now, I'm going to take you through the process, a thought process that every researcher would always go through. Now, what we are going to be doing is we are going to ask ourselves, what are the processes? What are these processes? What are the processes involved in genomic analysis? And then we'll see the answers that is going to bring out. Now, the reason why I'm doing this is for us to show us how sometimes when you are new to a field, how you are going to navigate your way into getting things done. Now, the first step is, of course, laboratory preparation and sequencing, sample collection, DNA extraction, library preparation, sequencing. Now, as a bioinformatician, your work starts after sequencing has been done. If you are not a bioinformatician, you are a molecular biologist. That is when you'll be talking about sample collection, DNA extraction. No, this one is the work of molecular biologists. Now, once the sequencing has been done, irrespective of the platform from which the sequencing was made, then you graduate to the bioinformatics analysis, and that is where your work comes to play. Now, there are different steps that are involved in genomic analysis as a bioinformatician. Number one is alignment. What you have to do with alignment is you are gathering your own data, you are now comparing it with other data on the database. And what does this inform? What does this give to you? It gives you an information about your own data. For example, now, how do we know that someone has a sickle cell disease? From their genomic data alone? From their genomic data alone, when we align it with a normal uh, person, a normal individual, the hemoglobin sequence. You know, hemoglobin is a protein, and sorry if this is new to some people. Hemoglobin is a blood protein that gives the blood, it assists in iron intake. Now, we now collect the hemoglobin, we sequence hemoglobin, like we sequence it, for example, and then we now take that of normal individual, we compare it with that of another individual that we do not know. Now, there is always a mutation in a particular protein of someone that is suffering from sickle cell anemia at a particular location. Now, when we compare the normal and the new sample that we have, and we saw that there is a mutation in the new sample, we can infer that that person has a sickle cell anemia disease, even without meeting that person. All we need is just the information about the DNA and everything. That is what alignment does for you. Now, the second stage is variant calling. You've now been able to compare your genome with that of the reference genome. What does that inform to you? You will now collect the different variants. You now compare the newly sequenced genome to the reference genome to identify differences or mutations. And that is what you call variant calling. And then you now annotate that variant. That is variant annotation. We have this background now. You can also proceed to interpreting and reporting and what have you and what have you. But now, from this little explanation, we've had an idea about what bioinformatician does when it comes to genomic analysis. Now, we'll now come back to this tutorial. So you can see the different array of what you can do as a genomic expert in terms of whatever you want to do. Now, we've seen genome annotation, so which is also one of the rules that the bioinformatician, a genomic expert, does. Now, if you scroll down also, you see methodology, assembly, genome assembly. You also have sequence analysis. You also have variant analysis. So these are things that you can do as a genomic expert. And that is why it is important for us to have that skill, hone it, and then use it for whatever it is that we want to do. Now, some questions will be asked that, okay, I'm studying this particular course. How does that affect, or how does that relate to my field? And these are some of the things that we do in a special training that we do for Galaxy for Africans. And it's a 60-day program that anyone that takes this program would have expertise in any field that they have chosen for themselves. Now, I'm going to take us through a process of genome annotation without using this Galaxy, because if we are to delve into it, but I promise, okay, let us do something. I'm going to give us as a bonus. Let us go through sequence analysis, which is one of the major ways or things that you have to do as a bioinformatician. So what kind of sequence that you want to analyze? What kind of sequence do you want to analyze? So you can check. There are different lessons that you can do as a bioinformatician that also relate to this particular area, sequence analysis. So one of such is quality control. How do you do quality control on your analysis? So here we are going to do an hands-on training, and we click on this and it will take you through a series of step-by-step guide on how to do sequence quality control. Now, why do you have to do quality control on your sequences? Now, let's go back to this particular area whereby we are talking about the step-by-step guide, sample collection, DNA extraction, library preparation, sequencing. Now, you've sequenced your sample. How do you know that the sample that you have sequenced is of utmost quality? How do you know? How can you guarantee or assure people or assure yourself that yes, I'm working with the best samples possible? I'm working with the best sequence possible. How would you assure yourself or the people that you are doing the analysis for or the result that you are presenting? If you are being asked, did you do quality control on your sample? And if as a researcher you are looking up, àìgbọ́fálà àwòkè as Yorùbás will say, you cannot, if you don't understand the Ifa corpus, that means you are just looking up and you are looking as if someone will tell you from up from the heavens. So you have to always make sure that the sample you are working with is of utmost quality, and failure to do that would jeopardize the result, whatever result that you are going to get in your analysis. And that is why it is important for you to always do that. Just give me 1 second to catch my breath and pump my system so that this thing will not die while we are still on our analysis. 1 second, please. Uh, so thank you. I'm back. So now we have our sequence. We want to do the sequence quality control. Now, when you go back to this particular, are we together? Let me confirm that before I go forward.
Yes, sir.
Yes.
Yes,
Good,
sir.
good. Thank you so much.
Yeah, we are together.
Yeah, thank you so much. So now, we want to do what we call the quality control. Now, there are different steps by step process that we have to take us through. The when you sequence your sample, you can either sequence your DNA or RNA. But as a genomics expert, there's nothing that concerns you with RNA. You are focused on DNA. Don't take that. Just take that literally. Everything concerns you with RNA. But for the purpose of this session, that's why I said nothing concerns you with RNA. Now, for each fragment in the library of your sequences or in the library, sequence is generated, and that sequence is called a read, which is simply a succession of nucleotides. Now, a DNA read can be 150 base pair long, depending on the type of sequencing that you do. Now, let me just do a little bit primer. There are different sequencing platforms available right now, and I'll tell you the most popular ones. The most popular ones are the Illumina, we have Oxford Nanopore, and we also have PacBio. Those are the most popular sequencing platforms available right now. You might have been hearing this Illumina, Illumina, Illumina. What does it mean? Yes, it means different one thing. You might have been hearing Oxford Nanopore. What does it mean? It has its own meaning. You might have been hearing PacBio. It has its own meaning, and they perform different functions. But Illumina is usually the most common one presently being used. Now, when you now have these modern sequencing technologies that generate massive number of sequencing reads in a single experiment, we assume that all these sequencing platforms, they are not perfect. And the amount of errors that is generated in each of these incorrect or imperfect technology has to be called. So, and this is due to the limitation in each of these technologies. So, it is necessary for us to identify and exclude error types that may impact the interpretation of our analysis. Now, another term that you have to be familiarize yourself is downstream analysis. Downstream analysis is just another word for analysis that you do for bioinformatics analysis. That's just downstream. So upstream, downstream is similar. So that means sequencing quality control is the first essential step that you have to take in your analysis. So because it will save you a lot of headache in the future. Now, one thing that we are going to do, the first step, there are one, two, there are inspect a raw sequence. There are different steps that you have to go through and that to determine the quality of your sequences. And yes, they are, it's a kind of a little bit long, and it will eventually give you the result that you are looking for if you do it properly. But I'm going to take us through just the first few steps so that to give us a taste of one of the things that we can do, and which would be a take home for attending this class, which is take a quality control, which is the most essential part. If your quality control is good, your other analysis is good. If your quality control is bad, your other analysis is going to be bad. So we are establishing a foundation that once it is well established, every other building that is being erected on top is going to come out strong and fine. Now, the first step is to create an history in your galaxy. And it is very simple. Um, where are you? Okay, let me open this. Let me use this. Let me use this. Let me use this. Let me use this. Okay. So we are going to create history. And how do you create an history on your galaxy? Come to this particular part and you will see this plus sign, which is create new history. You can click on create new history, and then you can come here, come down here to edit the name of the history that you want to create. So what name do we want to create? We will say, let's say Nobukimat, Nobukimat, Nobukimat quality control. So that's what we want to do, quality control. Then you can click on save. Once you click on save, and then you are good to go in this aspect. What's the next step? Now you can rename your history. We've done that. Renamed our history and we've done that successfully. The next thing is to import the data that you want to use. This time around, we are using a prepared data. If you are using your own data, there is another way that you are going to use that. So, but we are using a prepared data. So you can copy this link, and this data is from the data library and is a microbiome sample from a snake. A microbiome sample from a snake. That is what we want to use in this particular analysis. So what we'll go to do is go back to our interface, click on upload, and then come down to where it says paste or fetch data. Paste or fetch data. Now, if you are using a data from your laptop or your computer, you can choose local file. If you are using a data from the repository, you can choose from this repository. Now, there are different repository, but it's another field entirely that is not the focus of this session. So, but for the sake of this class, you click on paste and fetch data. And then here, in this platform here, Control V, and then after Control V, your the data, click on start, and then wait for it to load. Once this thing turns to green, click on close, and you will see the data is changing color here in this particular area, and you can see it's kind of orange. So you wait for it to turn green. Once it turns green, that means your data has already been uploaded here. Now, once you finish that, once that one is going on, you can come back here, and then uh, here you can check your data and see your data by clicking on the eye icon. So, and it's still loading. We are still going to do that. As you can see, once it's finished loading and changes to green, this eye icon, this view, you can see once you move your cursor to this area, it says view. You can click on view and then you can see the data once it has finished loading. But we are still going to do that. Now, it says that although it looks complicated, the FASTQ format is easy to understand with a little decoding. Now, Galaxy is teaching you what a FASTQ file means. Now, there are different file formats, which if we start to cover it, it will go beyond the scope of this particular session. There are different file formats that you'll be working with as a genomic expert. You have FASTA, FASTQ, GFF. You also have VCF files. So, but each of these file formats, they have their own peculiarities. So, and that is what we are going to be working with. So, but this one is FASTQ format. There are 4 lines in FASTQ. You have the lines that begin with @, the actual nucleotide sequence, the plus sign, and then string of characters. And this is how it looks like. The data, once it's finished loading, this is how it looks like. Let's check. Now it has finished. So click on the eye icon. You can see the data being displayed here. So you can see the ads, all this information. So from this information, you can see, you can detect everything that we are talking about in this aspect. Now we'll move on to the assessing the quality with FastQE. So now what you are going to do is to assess the quality. What you are going to do after this is to search for FastQE. FastQE. But if you want to do it in a fast way, I'll just click on this particular area, and it will take me directly to FastQE immediately. But let's do it and see what happens. Yes, I've clicked it. Once you click it, it will take you immediately to the tool. But if you want to search for it manually, you can search FastQE. Come to this area that are the right tools, and then you see FastQE. When you see FastQE, so you can see the information, and you click on this. Once you click on this, immediately it will take. But if you are doing a tutorial, once you click on the area, immediately it will take you directly to that aspect. But let me go back. Let me go back so that it won't distort what we were doing before. It won't distort. But I'm very sure it will distort the process that you have been doing. So, but let's click on this. Let's click on quality control. Let's go back to the tutorial. So that's why I opened it in 2 different area. So FastQE, let's search for it. FastQE in the tool section, and then click on it. When you click on it, it will automatically do the same thing. Now, what are the parameters you have to look for? FastQ data. In the FastQ data, click on reads. Let's scroll down. So in the FastQ data, once you click on the data set, you can select this data set because is this data set you want to work with. Let me cancel and reselect again. Multiple data set or data set. So when you click on it, you can select your data, and then it will automatically bring it here. So once that is done, score types to show, then main. Let's say score types to show. Let's leave it as main. And then you can click on run. So you can click on run, and then your data, you see the FastQE. So you can visualize your FastQ files with emojis. So just to see how it goes. So your data is already in queue. It will show here as it is being as it is loading. It will show here, but it's still loading. It will start, and when it is added to the queue already. So, and then the results will come out. You can see all the results. So now the next step is to do fast QC. Fast QC is to assess the quality. Or the fast QE is just a is just a bypass. So the real work that we want to do here is to check the fast QC. Now, as you can see, it has finished loading. Click on the eye icon to view your data, and you can see the data. That's this is just a kind of a game part. So you can see from the indication that the one that it looks like poop, that means it is rubbish data. So the one that is smiling, that is good data. So, but it's just by the way. Now we are going to search another tool, which is Fast QC. Fast QC, which is the actual work that we want to do. Fast QC. So Fast QC. Fast QC, once you click it, it will take it immediately here. And then what are the parameters to read? And then immediately you can just click on run. And then Fast QC is used for quality control. Now, Fast QC is used for quality control, and then you will see the raw data and then the web page. Then it will display the quality of your data in this way. If your data is, if this yellow things is within this green area, that means your data is of high quality. If your this yellow bar is in the red area, that means your data is of bad quality. So in doing that, you would know how quality your data is, and you know either to work with it or not to work with it. So it's as simple as that. So if you'd like to know more or do more analysis on this particular area, I've prepared other free tools for us that we can also run and also do successfully. So only those who will be a beneficiary, those that attended this session, are the only ones that will be a beneficiary of that free training. So if you have not registered your name or your email, please, this is the right time to do that. So I'll be sending it to you between today and tomorrow for you to work with, and then I'll be expecting you to send a feedback. And another benefit is that those that attended will be having a certificate of proficiency if they are able to submit their assignment within the time frame given to them. And I'll be looking forward to every one of you to do that. And while we are still waiting for um, the analysis to complete, there are other tools to do. Now that one is that we have seen that don't worry. If you are able to attend this session, automatically I know that we have your data stored already. So, or if you don't have it, you can send an email to noblekimat@gmail.com. You can send an email to noblekimat@gmail.com. Please help us type it in the comment section, noblekimat@gmail.com. If you not uh, maybe probably you are referred to the class session by a friend. So you can send an email to nobukimat@gmail.com so that you'll be sent the free tool that will make you do the analysis. Now, our analysis has finished running. So you can click on this eye icon to see the raw data. You can see the raw data of your data, and you can also see the web page. So, and I'm using a small screen. So if I'm using a big screen, it would have shown how it is in a big way. But let me download this data. Let me copy this link, and then let me check it out on another stuff. So let's see. It will download. Okay, let me just download. Let's see. So it has finished downloading. It will zip it as a file. And then let me extract. Let me extract all. Extract. And then you can view your results. You can view your result on your web page. HTML. So now it's much more bigger. So you can see that this data is more of it is in the red zone. And when it is in the red zone, that means your sequence quality is poor, very, very poor. So now the next stage is to trim, but there are ways in which you will trim and which would also be taught to us. So I would be looking forward to us submitting our information so that we can all be sent the data for us to conduct and then be sent our certificate of proficiency. So thank you for your patience with me for coming late to the session. And I'm very, very sure that we'll be seeing many, we're seeing ourselves a lot in the coming weeks. So thank you so much. If you have, I will give chance for one or two questions about what has been discussed so far so that every other information or inquiry will be discussed on the group chat. So thank you so much. If you have any questions, please don't hesitate to ask so that it can be given to you. Ask and it shall be given. So any question? Yeah, Mr. Clement Isibo, please go ahead.
Yeah, thank you for an excellent presentation. I needed to find out, can we do this with a session number from NCBI? Thank you.
Yes, yes. So you can download, you might not get it directly from a session number on NCBI. So, but you can download the data from NCBI and uh, upload it onto the in the repository. And I showed you three ways in which you can upload your data. You can either upload it from your computer, from the repository, or from the what's it called, from the fetch and paste data. So those are the three places in which you can upload it or you can upload your data from. So, but from a
Okay.
session number directly, you might not be able to get it from a session number.
The reason why I asked, because I have some transgenomic data which I wanted to do analysis on. So I don't know whether it's
Mm.
that's why I asked the question.
Yeah, you can upload it directly from the repository, but there is a way you will go through it, and there is a kind of data the way you will go through it. So SRA data majorly. So SRA data.
Thank you very much.
You're welcome, sir. I think there's another question. Let me check. Don't worry about the information on the assignment. It will be shared with us. But make sure that you registered so that you can have access to the information. Another question: how about those that are able to access to computer? Yeah, you can use your phone, but the best way is to have a computer so that you can have easy access to whatever it is that you want to do. So it's best to use your computer. Or you can use your phone if you are dealing with Galaxy. You can use your phone. Yes, there's no much difference for African Galaxy and use Galaxy in the sense that African Galaxy is dedicated for African African usage. So the server is dedicated for Africa majorly. So, Mr. Mohammed, your question.
Thank you very much for your wonderful presentation. I want to know what's the peculiar difference between NCBI and BLAST?
NCBI is the website. Blast is the tool on the NCBI website. So let me show you. This is NCBI. So NCBI has different platforms, different tools. So among the tools that is on NCBI is Blast. So let me see. So this is NCBI, the NCBI platform. This is Blast. As you can see, this is a popular resources Blast. So Blast is a tool on NCBI.
Okay,
So
thank you very much.
you can see when you click, you go to Blast directly. You're welcome. Any other question?
Hello, sir. Good afternoon, sir.
Good afternoon, ma. Asa.
Yes, sir. I mean, can we get the recording before the assignment so that it's easier for us to.
Yeah, the recording will be forwarded to you. And also to also ask to also answer the question of Miss Amina Omotoyos Balogun. Yes, it's confusing for someone that is new. But don't worry, as a bonus, I'll also be adding another fresher course for you to go through. But you have to make me a promise, that a special promise that I'm going to add also to the material sent to you. If you do not fulfill that promise, we are going to fight. So that means you have breaking our promise. But I don't want us to do that. But I'm going to send a fresher course to us. The question: what is the relationship of protein modeling and gene engineering? I don't know. I don't know the relationship between protein modeling and gene engineering. I don't know. So probably we can find out and see the difference between the two. So, any other
Yeah, good
question?
evening, sir.
Yeah,
Yeah,
Miss
good
Patricia.
evening. Okay. My question is, for someone coming from a non-STEM, right? In the aspects of biology education, and maybe you want to transit into bioinformatics and everything. First of all, everything seems so confusing, but not really confusing, but confusing. Then let me just put it like that.
Hmm.
So, what is your advice for okay for me, like how do I proceed? Like sometimes I find it difficult. Oh, you have to know a little bit of this, a little bit of that, a little bit of this. It's so challenging. And that thing again, I want to go like my master's into bioinformatics, but most times due to my undergraduate degree, if I make an application, they can say, oh, I don't have this and that. I find it challenging getting into um bioinformatics as for master degree. Is there something I'm missing, or is there a way I should channel it? Lastly, if I want to do like a full course with you, like there's something you mentioned about the 60 days, I don't know, 60 days course or something like that. Is there a discount, like a discount for people that are interested?
Yeah. Thank you so much. The first question is transiting into bioinformatics. So there are two ways to approach bioinformatics. One, you can approach bioinformatics by using bioinformatics as a tool in your discipline. Secondly, is to focus on bioinformatics itself. So the first one is using it as a tool. I presently use bioinformatics as a tool in whatever it is I'm doing. Not that I am saying I'm doing masters in bioinformatics. People do masters in bioinformatics, of course. But even if you do a master's in bioinformatics, so you still have to specialize in the area of bioinformatics, and that is one thing that is very cogent that we have to understand. So just, just a minute. So you use it as a tool as part of your discipline. Like now, I'm an entomologist, but I use bioinformatics in my field of entomology. So I'm a bioinformatician. I'm also an entomologist. So now that's solved that. If you are a microbiologist, you can use bioinformatics in your study of microbiology. If you are a zoologist, you can use it. So different aspects you can use. Now, the second part is focusing on bioinformatics as a course itself. So if you are a bioinformatician, you have to pick a discipline. You can't know everything about bioinformatics. It's not possible. It's not possible. So you have to pick a discipline. So transiting into it, it depends on what you are going into it to achieve. And we've done a series of webinar on that. So our operation managers will send us the recordings so that we can listen to it extensively and also to decipher a lot of things about that. So I think we'll have to call it a day so that we can quickly, because I'm still in transit, as you can see, I'm in the hotel lobby. So, but because I made it a promise that I'm going to, irrespective of whatever it is, I'm going to be here. So that's why we are having this session. So, God willing, I'm going to be sharing with us other resources that are necessary for our success and also for our journey into bioinformatics. So thank you so much and have a nice day. And I really appreciate everyone. And don't worry, the resources will be sent to us soon. So see
Thank
you.
you.
Thank you so much. It's a pleasure.